How will you define reproduction, children? Yes, the process of formation of young ones of a species by the grown-up individuals is called reproduction. Or it is the production of progeny possessing features more or less similar to those of parents. After all, reproduction is not necessary to maintain the life of an individual organism. Unlike the essential life processes such as nutrition, respiration, or excretion. If an individual organism is going to create more individuals, a lot of its energy will be spent in the process. In spite of this, organisms exhibit reproduction to increase the population and maintain the continuity of the species. Thus, reproduction is a vital process that gives more organisms with the same basic characteristics as their parents. It is required for the existence of life as well as perpetuation of the species. Also introduce variations. Reproduction at its most basic level involves making of the blueprints of the body design. Organisms look similar because their body designs are similar. If the body designs are to be similar, the blueprints for these designs should be similar. See the cow and the calf. They retain a basic body design. The blueprint of life is DNA, that is deoxyribonucleic acid molecules. DNA contains information for inheritance of features from parents to next generation. The DNA in the cell nucleus is the information source for making protein. If the information is different, different proteins will be made, and these different proteins will eventually lead to different body designs. Thus, a basic event in reproduction is the creation of a DNA copy. Cells use chemical reactions to build copies of their DNA. This creates two copies of the DNA in a reproducing cell. They will need to be separated from each other. However, keeping one copy of DNA in the original cell and simply pushing the other one out would not work because the copy pushed out would not have any organized cellular structure for maintaining life processes. Therefore, DNA copying is accompanied by the creation of an additional apparatus. The DNA copies separate each with its own cellular apparatus. Effectively, a cell divides to give rise to two cells. The two cells are similar, but the characterization of cells depend on the copying reactions because no biochemical reaction is absolutely reliable. The process of copying the DNA will have some variations each time. As a result, the DNA copies generated will be similar but may not be identical to the original. Some of these variations might be so drastic that the new DNA copy cannot work with cellular apparatus. Such a newborn cell will simply die. The surviving cells are similar to but subtle different from each other. This inbuilt tendency for variation during reproduction is the basis for evolution. Let us know the importance of variation. Reproduction is linked to the stability of populations or species. Populations of organisms fill well defined places or niches in the ecosystem using their ability to reproduce. The consistency of DNA copying during reproduction is important for the maintenance of body design features that allow the organism to use that particular niche. However, niches can change because of reasons beyond the control of organisms. Temperatures on the earth can go up or down, water levels can vary, or there could be meteorites hits. If a population of reproducing organisms were suited to a particular niche, and if the niche were drastically altered, the population could be wiped out. So, variation is useful for the survival of species over time. If some variations were to be present in a few individuals in a population, there would be some chance for them to survive. For example, if there were a population of bacteria living in a temperate water, and if the water temperature were to be increased by global warming, most of these bacteria would die. But the few variants resistant to heat would survive and grow further. That means variation is useful for the survival of species over time. Okay? There are two main modes of reproduction in organism, that are asexual reproduction and the sexual reproduction. The mode of reproduction in which new individuals develop from specialized or unspecialized somatic part of a single parent is called asexual reproduction. Let us study modes of reproduction used by single organisms. The usual method of reproduction found in single organisms is asexual reproduction. It occurs by fission, budding, fragmentation, regeneration, spore formation, and vegetative propagation. Fission. It occurs in unicellular organisms. The parent cell divides into two or more cells which function as independent individuals. Hence, the identity of the parent is lost. Depending upon the number of individuals formed, fission is of two types: binary fission and multiple fission. In binary fission, a mature unicellular organism, for example, amoeba, divides into two daughter cells. For binary fission, the nucleus first elongates and then divides into two. It is followed by constriction of the cytoplasm between the two nuclei to form two daughters. In amoeba, binary fission can occur in any plane. However, some unicellular organisms show somewhat more organization of their bodies. In Leishmania, a protozoan which cause Kala azar or black fever, which have a whip-like structure called flagellum, and it undergoes longitudinal binary fission. Means binary fission occurs in a definite orientation. Other single-celled organisms such as the malarial parasite, that is Plasmodium, divide into many daughter cells simultaneously by multiple fission. Multiple fission, a mode of fission in which a single unicellular individual produces a number of daughter cells. This occurs due to repeated divisions of its nucleus followed by the cleavage or division of cytoplasm to form uninucleate daughters. They are released by the bursting of the parent cell. Another unicellular organism, yeast, on the other hand, can put out small buds that separate and grow further. You may see green filamentous structures floating in pond water. This green filamentous structures you see are an algae named Spirogera. In Spirogera, each filament has a single row of cylindrical cells and each cell has spiral bands of chloroplast. When a Spirogera filament breaks into pieces, each piece grows into a new filament by cell division. This process is called fragmentation. During this process, the body of an individual breaks up into two or more parts and each part develops into a complete organism. In complex organisms, all cells are not similar. The cells are organized into tissues and tissues into organs. The different organs are placed at different positions. If such an organism breaks off at any point, the broken part cannot grow into a complete organism with all organs. A basic strategy used in multicellular organisms is that different cell types perform different specialized functions. Reproduction in such organisms is also the function of a specific cell type. Such specialized type of cells are capable of growing, proliferating, and making other cell types under the right circumstance. Next is regeneration. It is the ability to grow parts lost in an accident or injury. In extreme forms of regeneration is found in planaria. The animal can be cut into many pieces, with each piece growing to form the complete animal. Here, regeneration is helping in asexual reproduction. Regeneration is carried out by specialized cells. These cells proliferate and make large number of cells, and from this mass of cells, different cells undergo changes to become various types of cells and tissues. These changes take place in an organized sequence referred to as development. Most animals do not undergo regeneration for the purpose of reproduction, as no organism will wait for being cut in order to multiply. Regeneration is usually meant for repair of injured areas or parts lost accidentally. For example, tail of wall lizard, arm of starfish. Okay. Sometimes new individuals develop from the body wall of the parent as bulb-like projections called buds. The buds may be unicellular or multicellular depending upon the type of parent organism. Organisms such as Hydra use regenerative cells for reproduction in the process of budding. In Hydra, a bud develops as an outgrowth due to repeated cell division at one specific site. These buds develop into tiny individuals and, when fully mature, detach from the parent body and become new independent individuals. Dear children, we can continue with asexual reproduction. You might have seen gardeners taking cuttings from the stem of a rose plant and planting them in the soil under suitable conditions. Then these cuttings grow into new rose plants. That is, the vegetative parts of a plant, such as the root, stem, leaf, etc., can produce new plants. And this process is called vegetative propagation. Vegetative propagation is common in plants like orchids, ornamental plants, and grasses. Plants such as banana, rose, jasmine, etc., which do not produce seeds, can be grown by vegetative propagation. Next, artificial modes of vegetative propagation. Farmers and gardeners have developed various artificial methods of vegetative propagation like grafting, layering, cutting, and tissue culture. Cutting is a very simple method of propagation in which a piece of the parent plant stem with nodes and internodes is placed in moist soil. This grows into a new plant. In grafting, the cutting of a plant is attached to the stem of a rooted plant. The attached cutting becomes a part of the rooted plant and draws nutrition from it and grows further. In layering, one or more branches of the parent plant are bent close to the ground and covered with a moist soil. The covered portions grow roots and develop into new plants. Okay. Do you know what is tissue culture or micro propagation? In this technique, some cells or tissue from a desired plant is placed in a suitable nutrient medium under proper conditions. The tissue grows into an unorganized mass known as callus. The callus is transferred to another medium containing hormones for growth and differentiation. When the plantlets grow, they can be transplanted in the soil or in pots for developing to maturity. Tissue culture allows us to grow a whole plant from cells taken from any part of the plant body. Many plants can be grown from one parent plant in the laboratory under controlled disease-free conditions. This technique is commonly used for ornamental plants. Let us see the advantages of vegetative propagation. The plants raised by vegetative propagation can bear flowers and fruits earlier than those produced from seeds. The propagation of plants such as banana, orange, rose, and jasmine, which have lost the capacity to produce seeds, can propagate by vegetative propagation. Then, all plants produced by this method are genetically similar. Let us see the activity 8.5 on page number 132. Take a potato tuber. Observe it carefully. It has a number of notches. When viewed with magnifying glass, the notches are found to have buds. Cut the tuber into two types of pieces. One with notches and the other without notches. Place them alternatively on a wet cotton kept in a tray. Sprinkle water from time to time. After about 4 days, the tuber pieces with buds are seen to have developed plantlets. The other pieces shrivel and start decaying. The fleshy leaves of bryophyllum bear adventitious buds in the notches along the leaf margin. These buds develop into small plants under favorable conditions. These plantlets can be easily separated to grow as independent plants. Next, activity 8.6 on page number 132. Cut two pieces from a money plant, one with leaves and the other without leaves. Place these with one end immersed in water, kept in a transparent bottle. Leave them like this for a week. You will see that roots and new leaves grow on the piece with leaves, while the other piece gradually decay. This is because a plant can grow new leaves and branches only if it has nodes. New leaves and branches arise at the nodes. The piece of money plant which does not have nodes cannot grow because it cannot produce new leaves. Okay? Take a piece of moistened bread and keep it inside a polythene bag for 2 to 3 days in a warm, humid place. You will observe yellow and black patches on the bread slice. You will see a powdery substance with spores. The spores are of bread mold or rhizopus. The molds also have thread-like structures called hyphae, through which they draw nutrients from the bread. They are not reproductive parts. Another asexual means of reproduction is spore formation. Spores are asexual reproductive bodies enclosed in a thick-walled structure called sporangium. This sporangium can tide over unfavorable conditions such as extreme heat, dryness, acidity, and so on. Spore formation is a common method of asexual reproduction in many lower forms of life such as algae, bacteria, and fungi. Under favorable conditions, the spores are released by the breaking of the thick wall of the sporangium. The spores then germinate into new individuals.